everyone. Welcome to the Twisted DNA. So the topic for the day is gel electrophoresis, and in this video we're going to start by defining gel electrophoresis and know some of the requirements. Then we'll talk about how does a gel electrophoresis work, and last we'll also look at an example. So let's start by asking a question. What does gel electrophoresis mean? The word phoresis mean movement or migration, and the prefix electro means there is a use of electricity for the movement to take place. So as the name suggests, gel electrophoresis is a laboratory method that utilizes gel to separate mixtures of DNA, RNA, or proteins according to their molecular size and charge. So, what are the requirements for gel electrophoresis then? Well, first one is the gel, of course. Second is voltage supply. We also need buffer. And last but not the least, we need loading die. Now, let's talk about each of them in detail. The first one is gel. So you might be wondering, can any gels be used for the electrophoresis technique? Unfortunately, the answer to that question is no. Well, in research labs, agarose and polyacrylamide gels are the most commonly used gels in electrophoresis. Agarose gels are commonly used for the separations of nucleic acid like DNA or RNA, whereas polyacrylamide is the preferred gel system for separating proteins. For the sake of this video, we will focus mainly on agarose gel electrophoresis. So now we know that in agarose gel electrophoresis, we use agarose to make gel. So what is this agarose then? Well, agarose is a natural polymer prepared from red algae, and agarose can be dissolved in buffer, and a gel can be formed by cooling it at below 45 degrees Celsius, and it forms a gel as a result of extensive hydrogen bonding between agarose chains, and a tiny pores would be formed in a gel, which would look something like this on the right. So the pore size of agarose actually depends on the agarose content. Higher the concentration of agarose, smaller would be the pore size in the gel. So agarose with large pores would allow the movement of very large molecules, and the vice versa is true. For example, here the pink sample is larger than the yellow one. So the pink sample gets trapped in the mesh and takes more time to travel, whereas the yellow sample has no trouble moving through the gel matrix due to a smaller size. So the second requirement is the voltage supply. And as I have mentioned at the beginning of this video, that electricity is required for the separation of our sample. So now let's talk in some detail about why the electric field is so essential for the separation. So when the voltage is applied during electrophoresis, it creates this electric field such that on one end of the gel, we have a positive charge and the other end has a negative charge, just like the one shown in this picture. So DNA and RNA are negatively charged because of the phosphate group that is present in their backbone. Therefore, when an electric current is applied to the gel, DNA and RNA can move towards the positively charged electrodes, which allows for the separation. However, for proteins, they're not negatively charged. So when we want to separate proteins using gel electrophoresis, we must first mix the protein with some detergent, which we will talk about more in our next video. So the third requirement is the buffer. So buffer is basically water with some salts added to it so that it can resist the change in pH. And buffer in gel electrophoreses are used to provide ions which are necessary for the passage of currents through the gel, and this allows to maintain a constant pH throughout the gel electrophoreses. By maintaining this neutral pH, we control the net charge of our sample, which helps in proper migration and separation of our sample. Tris-borate EDTA or TBE and tris-acetate EDTA or TAE are the most commonly used buffers in agarose gel electrophoreses. So how do we know which buffer to use then? Well, TBE is actually a good choice when you need high resolution for small DNA fragments, whereas TAE is a good choice when you're working with large DNA fragments. Note that it is very important to use the same buffer that was used to dissolve your agarose. So the last we have is loading dye. So after the DNA has migrated through the gel, it needs to be visualized. While DNA or RNA are colorless, so we need some kind of loading dye in our sample that will help us determine the rate of movement of different size molecules during the gel electrophoreses. So now that we've covered the requirements for agarose gel electrophoreses, let's look at how does an agarose gel electrophoreses actually work. So for a typical agarose gel electrophoresis procedure, the gel matrix is cast in a slab. This is done by dissolving the agarose in a buffer, and plastic combs are used to create wells. And these wells are important because they provide a place for samples to be loaded. And before loading the sample, it is important to mix the sample with the loading dye, as these dyes weighs down the sample and make sure that the sample does not leave the well. And loading dye also provides this visible marker which helps us to track the progression of the run. Oftentimes, a ladder is used alongside the sample, and these ladder contains the known lengths of either DNA or RNA, which can be used later on for size comparison. The gel is then placed in a gel tank and filled with buffer solution, and an electrical field is applied along the length of the gel. And after the run is complete, a gel is stained with a fluorescent dye such as ethidium bromide and placed under UV light so that the DNA or RNA fragments can be observed. And a band with high intensity in the gel indicates that there is a higher amount of DNA or RNA present compared to the band of less intensity. So let's take a look at an example. So this over here is what somewhat the actual gel image would look like. Here at the top are the wells where the samples are loaded, and in the first well that says M is where DNA ladder or marker was loaded. And we can see that there are several bands, and the sizes of these bands are already known, and these sizes are also labeled here in the picture. And now we can compare our three different samples with this marker and determine the length of our sample and make sure whether we have the right size sample or not. So here we can compare that sample 1 has about 600 base pair DNA, and the reason we know this is by comparison with a marker. Similarly, the sample 2 has about 500 base pair of DNA sample, and note that the 500 base pair DNA sample is smaller than 600 base pair, and this 500 base pair sample has traveled further than the sample 1. And if we look at sample 3, we can see there's like 2 different bands. So this tells us that there are 2 different types of DNA molecules, and these DNAs are of different sizes, as we can see that these are separated so perfectly. So to summarize everything that we have discussed so far, so gel electrophoresis is a method to separate different molecules with the use of electric field. The concentration of agarose determines the pore size, and the larger pore size allows larger molecules to travel through the gel, and vice versa is true. And we also need some kind of loading dye to visualize our results. So that's it for this video. I hope you like it and hope it wasn't too long. Please don't forget to like, comment, share, and subscribe, and I'll see you all next time.